Hello and welcome back to another biology episode. Today we're talking about autophagy, which is a process to degrade and recycle intracellular material. It's a fact that cells within our bodies have to be constantly repaired and remodeled, and therefore it's necessary to recycle intracellular material. And we use the term autophagy to describe the delivery of cytoplasmic material to the lysosome within animal cells. Lysosomes are organelles which contain a lot of enzymes that are required for the breakdown of, for example, proteins. So autophagy is a highly regulated process to sequester misfolded proteins, damaged or aged organelles, but also mutated proteins within vesicles, which we call autophagosome. So we not only have to remove and recycle cellular content, but it's also required to sustain homeostasis. The word autophagy originates from the Greek words auto for self and phagy for eating. And if we have detrimental environmental conditions like the lack of growth factors, infections, or also starvation, then autophagy gets induced. Furthermore, cytokines and hormones like cytosol, which is depicted on the right side, can regulate and initiate autophagy. We have actually three major types, which are macro and microautophagy, but also chaperone-mediated autophagy. The term autophagy in general predominantly refers to macroautophagy. Microautophagy is the process where the contents of the cytoplasms enters the lysosome through deformation of the membrane or through invagination, so more or less directly. Knowledge about this process is at the moment limited. Then when we're talking about macroautophagy, its exclusive feature is that the membrane gets sequestered not from the membrane of the lysosome, but a vesicle-like autophagosome is formed de novo. Chaperones are proteins that are required to help other proteins to fold correctly. It's a highly regulated and specific process when we're talking about chaperone-mediated autophagy, and around a third of all proteins have a sequence that is required to get unfolded by chaperones, and they then get degraded in the lysosome. The process of autophagy is highly conserved from yeast to humans, which highlights its importance. Autophagy-related genes are denoted as ATG. Over 30 have been described so far, 18 of which are necessary for the core function. 4 critical steps are required for the process of autophagy. We have initiation, nucleation, maturation, and degradation. All of these steps include various proteins. You see an example depicted here, but they cannot be discussed in detail. Let's just talk about the principle. Autophagy begins with an isolation membrane, which is also known as phagophore. This membrane might originate from mitochondria, plasma membrane, or also the endoplasmic reticulum. The phagophore then expands to engulf intracellular cargo, which can be protein aggregates, organelles, or ribosomes. So thereby the cargo gets sequestered in a double-membraned autophagosome. And this loaded autophagosome then majored through the fusion with the lysosome, which you see here. This promotes the degradation of autophagosomal contents by proteases, so enzymes. A video about enzymes is linked above. By proteases, which can break down proteins. Lysosomal permeases and transporters then export the amino acids and also other byproducts of the degradation back into the cytoplasm, where they are reused for building macromolecules and metabolism in general. So autophagy can be thought of as a cellular recycling factory, and it's a physiological process. Therefore, it's required to maintain healthy working cells and is also a survival strategy for a cell. Furthermore, it's essential in regulating cellular homeostasis, so dysregulations have been found in various pathophysiological contexts, like neurodegenerative diseases, cardiomyopathy, infectious diseases, type 2 diabetes, fatty liver, but also cancer. this was a quick overview of the process of autophagy. If you enjoyed this video, don't forget to give it a thumbs up, subscribe to the channel to see weekly uploads, and see you in the next video.